Okay, so we're looking at examples of cell signaling. I'll probably have to put this video into two pieces, but here we go. You should be familiar with just the general information in terms of apoptosis, regulation of the cell cycle, and how it's associated with cancer, as well as development. Development is new, so definitely take a look at that if you're confused. All right, so for apoptosis, we went over this in class. Programmed cell death, you've got a signal, transduction, and response. That's our big idea, right? If we're thinking about examples of cell signaling, then we need to have that transduction and response. So the signal itself may be either external or internal. You can think about improper DNA replication, or anytime the ER isn't properly folding the protein, so you have a whole bunch of misfolded proteins, that can also send the signal. In C. elegans, we can look at the SED proteins, but you don't really need to know the mechanism of how those are activated. You just need to know that somehow the signal is transduced to create the response. The response you will need to know, which is going to be the activation of nucleases and proteases. So we're breaking down the nucleic acids, the proteins. An important protease that's involved is caspase, and that is always present but not active. It's not going to be activated until you get the signal. In terms of the result of that, what you end up with is the cell becoming kind of blebbed off and fractionalized, and it's really important that we then have another cell that can recognize the signals of a dying cell that can then come in and engulf those parts so that they don't just leak out all over the place. There's all those enzymes in the cell that are really dangerous to get them uncompartmentalized, right? They're in specific areas so that the rest of the cell can stay safe. If we just leak those all over the place, the neighboring cells are going to have a lot of damage too. And so when you just have normal cell death, for example, if you go outside today and stand out there for too long, then the cells will start to die, and that necrotic cells, those necrotic tissues, are going to also cause damage to nearby cells. Whereas apoptosis, you're not going to have that happen. So as I just mentioned, we've got the signal molecule, in this case a death signaling molecule. How cool does that sound? We've got our activation cascade of the various SED proteins so that you can have the proteases and nucleases become activated. Again, you don't need to know very specifically what's going on in C. elegans. It's just kind of the example that the textbook used, although you do have other SED proteins in your cells ready to go for apoptosis if need be. So why waste your time making a cell? Why waste your energy making a cell if it's just going to be destroyed anyway? Obviously, if there's any kind of cancer or something's gone wrong in cell division, it's going to be important that we can stop that ahead of time. And so that's going to be a big role of apoptosis, but also in development, making sure that your fingers and toes don't end up all webbed. Various animals don't have a lot of apoptotic mechanisms in these finger tissues, so they end up with much more webbed fingers. You know, you can think about ducks and their little feet. There's obviously a clear reason to keep the webbing there, but for us that would be a wee bit ridiculous, so we get rid of that. Also in your immune system, but we'll talk about this more later in the unit. Okay, we know how the cell cycle is controlled. There's major checkpoints at G1, G2, and M, and those checkpoints are really just a way of checking in on what chemicals are active, and if the correct combination of chemicals are active and deactive and present or not present, like you have the right combination of stuff there, then the cell can go ahead. If you don't have the correct set of stuff there, then that checkpoint acts as like a stop, and you can't go beyond that. So for example, if DNA replication doesn't happen properly, then as it's going through the cell cycle, the G2 checkpoint is going to ensure that all of that DNA has been properly replicated. If it's not, then you're going to have different chemical markers present in the cell that is kind of like a way for G2 to check. I'm doing air quotes right now, you can't see me, but whatever, to check that everything is good there. So good stopping points, G2, and through as mitosis is going, you've got the M and then G1. Typically, if you can get through G1, you're going to do okay going through the rest of the cycle, but that's not always the case. And G1 is also going to be that like exit ramp into G sub 0, or like the inactive cell phase. So at that point, the cell is no longer in the cell cycle. It is just chilling, doing its normal things. You can think about nerve cells and so forth. Okay, so there's all kind of external chemical signals that affect the cell cycle, such as growth factors, density-dependent inhibition, and anchorage dependence. A chemical marker of cancer is that the density-dependent inhibition doesn't really stop mitosis from happening in cancer cells. Same for anchorage dependence. So density-dependent just is cell-to-cell signaling. If a cell is touching another cell, it is not going to continue to grow. If it is not touching other cells, it may promote growth. For example, if you have an injury, the cells that are now exposed on one side will start dividing to fill in that area. Anchorage dependence just means that you're stuck on something else. So some kind of substrate is usually what we refer to, either other cells that are underneath of it or like underlying tissues. So that attachment to the substrate is going to allow for cells to continue to divide. If they're just kind of floating off in space, then they're not going to have that same process happen. There's obviously examples where this isn't quite true, such as different types of blood cells, but otherwise that's generally the case. You can also have internal chemical signaling, and the big one that we talked about in class is CDK, cyclin. Cyclin is what the C in CDK is, so cyclin-dependent kinase. When you have the two of these chemicals combined, you end up with a maturation promoting factor. And so an MPF is a kinase that just depends on the presence of cyclin. So when cyclin levels are high, we're thinking they're rising in S, they're rising in G2. When the cyclin levels get high enough, the CDK can become activated to make MPF, and MPF is going to do a whole bunch of stuff to allow mitosis to progress. So it's going to be phosphorylating proteins involved in things like the breakdown of nuclear envelopes, allowing the chromosomes to condense and stabilizing the spindle fibers. In other words, all of those things that have to happen in mitosis, not all of them, but a lot of them, are going to depend on the presence of MPF. So if your cyclin level hasn't risen sufficiently during S and G2, then you're not going to be able to progress into M, into mitosis. And then during mitosis, the cyclins get degraded, and that's going to prevent a cell from just like continuing with dividing. So we already touched on this. What is cancer? It's the uncontrolled cell division. So the checkpoints aren't working. There might be something wrong, and the checkpoints that should stop it don't. And external signals to stop dividing or to commit apoptosis are being ignored. Now, just that uncontrollable cell division is only really a tumor, and it's not until the tumor breaks away from everything else that we're going to think about malignant cancers. So malignant meaning bad, benign meaning good, even though no one really wants a benign tumor, but it's not really like killing you right off the bat. Benign tumors can just be things like fatty deposits, although if they're in a bad spot, then certainly they can cause damage on their own. But malignant tumors are those that have already broken away from the substrate, so they don't have that anchorage dependence, and they're just floating around, and then they can like adhere to another area and start growing. So that's metastasis, right? So maybe you have, let's say, a liver cancer, and so that chunk of liver tissue, of cancerous liver tissue, is going to float around until it ends up somewhere else, maybe clogged in your heart, and just starts growing there instead. And so, you know, that sucks. And that is our definition of cancer. It's when you have a tumor, right, that uncontrolled cell division, and it has metastasized. It has broken away from the rest of the tumor area. Okay, so two internal signaling pathways for controlling the cell cycle are going to be the proto-oncogenes and the tumor suppressor genes. Proto-oncogenes are normal. Those are good. It's when they become mutated that they're oncogenes. One common example that we looked at here is RAS, and when RAS is stuck to GTP, it just stays active. So that means it's an oncogene. It's activating a transduction cascade that causes the promotion of the cell cycle. Tumor suppressor genes, on the flip side, those are going to normally produce proteins involved in the inhibition of the cell cycle, but if they don't work properly, like if the gene is mutated and they're just not there, then there is no stop signal when things kind of go all to hell. So we can take a look here at both of those. We've got an RTK, receptor tyrosine kinase. The growth factor is activating the RTK, which is activating the G protein. So if we have RAS just constantly on, that GTP is stuck there, then the growth factor can yeet out of there. The RTK can become deactivated, but that RAS is still sending a signal even beyond when the growth factor is actually there. So it's as if the growth factor stays there, sending the signal grow, 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 and that signal doesn't get turned off. So the RAS mutation just causes the constant flow of phosphorylation and activation of transcription and gene expression, and then the cell cycle is just doing its magical thing, even though there are no growth factors present anymore. So that's, sucky, no bueno. And then on the P53 side, we can take a look at the normal pathway here is that there's some kind of damage. So this internal signal of DNA damage, or there's a number of other things that it can be caused from, but we've activated P53. P53 comes in and it's like, okay, this is no bueno, and that's going to inhibit the production of the proteins that are usually involved in promoting cell signaling, right? So, sorry, not cell signaling, cell division. So we've shut that down and life is good. But if we're not making active P53, then DNA damage doesn't matter. It's just going to keep going through the cell cycle, ignore the checkpoint. The G2 is where it would be noticed, and that's when P53 should be made to prevent movement into mitosis, but instead it just goes right on into mitosis. Damage, no one cares, right? So those things combined are going to allow cancers to start happening, right? You start forming the tumor, and then something having to do with failure to properly anchor is going to allow that tumor to metastasize. So I am going to go on into development in the next video. If you've got questions, you know the routine, and enjoy your snow day.